personal congratulations to you all on achieving your awards today from Liverpool John Moores University, LJMU, in association with YPC. Everyone is very proud of your achievements and are delighted with your success.
Exciting, it's very exciting.
Saya rasa gembira dengan kejayaan anak saya berjaya menamatkan pengajian di YPC dan hari ini adalah hari yang paling gembira lah pada saya kerana di atas kejayaan.
Very excited, super excited actually.
Kind of feeling happy, very happy.
It's a great honor that I can graduate with Liverpool John Moores University degree.
I can invest my career in multiple spectrum. I am more versatile for all the challenges that I have to face.
After graduation, I'm going to continue working and explore the world with my current company. Other than going to Japan, we're also looking forward to expand the team to Singapore and as well as other countries in Asia.
Liverpool John Moores University itself, a 200-year-old university that is really world-class and world ranking. And what does it do to you? It opens the door to future job employability, not only in Malaysia but also all over the world.
YPC International College is a very well-respected partner at Liverpool John Moores University. We're very glad to see another big cohort of students graduating today. Congratulations.
Baiklah, Assalamualaikum dan selamat sejahtera kepada pegawai-pegawai PPD, semua pelajar, guru-guru dan ibu bapa yang menghadiri seminar Jom Skor STPM 2023 bagi semester 3 bagi mata pelajaran Biologi bersama Mr. T. Program ini merupakan jaringan kolaboratif strategik YPC International College bersama Kolej Tingkatan 6 Pudu Jaya. Anda bersama dengan saya, Cik Laila, selaku pengerusi majlis pada hari ini. Bagi pelajar online, kalau boleh dengar suara saya dengan jelas, minta tolong respon di ruangan chat box. Baiklah, terima kasih kepada yang beri respon. Bersama-sama dengan kita pada hari ini adalah yang berusaha, Puan Aminah binti Daud, Pengetua Kolej Tingkatan 6 Pudu Jaya, Puan Sarinah binti Yusof, Guru Penolong Kanan Tadbir dan Kurikulum Kolej Tingkatan 6 Pudu Jaya, serta guru-guru dari Kolej Tingkatan 6 Pudu Jaya. Mr. T, Guru Mata Pelajaran Biologi, Datuk Dr. Noor Robert, Pengetua YPC International College, Dr. Farah Robert, Pengarah Urusan YPC International College, Kaunselor Akademik YPC. Seterusnya tetamu jemputan kita pada hari ini iaitu pelajar-pelajar STPM dari seluruh Malaysia. Terima kasih juga kepada ibu bapa dan guru-guru yang turut hadir pada petang ini. Sebelum memulakan agenda utama kita, Sebelum memulakan agenda utama kita, suka untuk saya ingatkan beberapa perkara terlebih dahulu. Pastikan mikrofon dalam mod senyap. Yang kedua, pinkan nama wakil yang akan berbicara sebentar lagi untuk melihat slide pembentangan. Pastikan anda bersedia bersama buku catatan untuk mencatat isi-isi penting. Dan yang terakhir, sebarang pertanyaan boleh terus bertanya di ruangan chat semasa sesi Q&A. Diingatkan kepada semua pelajar untuk tunggu sebentar setelah kelas tamat bagi mengisi kehadiran untuk tujuan penyerahan sijil penyertaan. Sebelum itu, semua pelajar dan guru-guru dijemput untuk follow dan subscribe media sosial YPC International College di Instagram, Facebook, TikTok dan YouTube kami untuk mendapatkan info-info program akan datang. Baiklah, bagi memulakan program kita pada hari ini, saya ingin menjemput Miss Aminah, pensyarah daripada YPC International College, dengan segala hormatnya dipersilakan.
Okey, terima kasih Miss Laila. Assalamualaikum dan salam sejahtera kepada pengetua PKM, penolong pegawai tadbir, guru-guru Datuk Dr. Noor Robert, Dr. Farah, YPC and LJMU staff, para ibu bapa dan adik-adik yang join program kita pada hari ini. So hopefully semua dapat dengar suara saya dengan jelas ya. Okey. Pertama sekali, izinkan saya memperkenalkan diri saya. Nama saya Puan Aminah. Saya merupakan seorang lecturer dan juga program coordinator di YPC International College. Sebelum itu saya nak ucapkan selamat berpuasa kepada semua yang berpuasa, minggu pertama puasa untuk minggu ni. Okey. Pada hari ini, saya ingin berkongsi sedikit tentang one of our most popular and in-demand program offered by YPC. So, program yang akan saya kongsikan pada hari ini merupakan degree program from Liverpool John Moores University UK, iaitu Bachelor in Science with Honours in E-Business Technology and Management, atau singkatannya we call it as a BEM lah. Okey, untuk makluman adik-adik, this degree is a multidiscipline. Maksudnya dia combine two areas of study. So untuk BEM, it covers IT dan juga business. So if adik-adik minat business but on the same time adik-adik nak dapatkan kelebihan dalam bidang IT, BEM adalah pilihan yang paling tepatlah untuk adik-adik semua. Kerana melalui BEM, adik-adik dapat mempelajari front-end aspect of e-business seperti digital marketing and sale, as well as back-end IT aspect seperti web development and database management. Okay, some of the subject yang adik-adik akan belajar dalam program ni termasuk e-commerce management, website design, database design, system design dan juga software development. Okay, untuk makluman adik-adik, this skill are very essential for anyone yang ingin berkecimpung dalam bidang e-business industry, whether as a business owner ataupun professional in a technology company. Apa yang menarik lagi adik-adik, our certificate ni adalah UK degree certificate. Bermaksud our certificate ni bukan hanya diterima di dalam negara tetapi turut diterima di peringkat antarabangsa. Contohnya seperti di UK, Japan, Thailand dan Dubai. Okey, untuk adik-adik yang ada cita-cita nak bekerja dekat luar negara, so ini adalah peluang adik-adik untuk mencapai cita-cita adik-adik semua. Okay, if adik-adik berminat dengan program ni, di sini adik-adik untuk lepasan STPM ni, adik-adik boleh menyambung ke degree level selama 3 tahun sahaja dan adik-adik boleh apply dengan menggunakan result trial STPM. Dan jika adik-adik berminat dan nak mengetahui info dengan lebih lanjut, adik-adik boleh cek pada ruangan komen dan our counselor will assist you lah. Okay, di sini saya ingin berkongsi sedikit tentang our alumni and what career they had secure as a BEM student. Okay, di sini adik-adik boleh tengok kat sini kita ada 3 orang alumni yang sekarang ini bekerja di luar negara seperti Gita, Wenlin dan juga dan juga Navin. So untuk Gita, currently she is a principal consultant at Manhattan Associates Singapore. For Wenlin, he is a senior account executive at MAT Thailand. For Navin, he is currently international sales manager Amazing Fruit Trading at Dubai UAE. Okay, untuk Gajendra Gurunathan, he is currently a senior IT analyst at Deloitte. Deloitte ni adalah multinational company based dekat UK, England. Okay, untuk Sukhbir Singh, currently he is a global payment manager at Zil Group. Dan for Intan Baiduri Kamalul Bahri, she is a senior system support at Jaya Sompo Insurance Berhad. Okay, we go to the next slide. Okay, ini adalah our alumni yang graduate Julai tahun lepas. Kalau adik-adik yang tengok kat sini, Ho Sochet and Tan Yuan Yi, they are more towards IT, which is Ho Sochet kali ni is a software engineer dekat Dina France Systems Sdn Bhd, and Tan Yuan Yi is a front-end developer or programmer at TDC at Malaysia. Dan untuk yang lain, seperti Physician, she is a service marketing executive at Hudson Star Sdn Bhd, and Kon Hua Hen, currently he is a QA/QC specialist at Flash Express Malaysia, and Ellie Lim Yitan is a procurement at Genting Malaysia Berhad. Untuk si Mayton pula, she is a HR executive at Vertet Mastery. So most of them dapat kerja dekat company yang agak well established juga. Okay, berbalik kepada program kita pada malam ini, saya ingin menjemput guru yang akan mengajar pada petang ini adalah Mr. T yang merupakan guru daripada KTE Pudu Jaya. Beliau sudah berpengalaman selama 15 tahun dalam mengajar. Jadi saya ingin menjemput Mr. T. Dipersilakan Mr. T.
Okay, first of all, okay, I would like to thanks to YPC International College for giving me this opportunity to share some of the answering techniques in STPM Biology for semester 3. Okay, so without wasting time, let's revise the format of assessment for this Biology Paper 3. Okay. Paper 3 consists of 3 sections, okay, same as your paper 1 and also a paper 2. Okay, section A, 15 marks were allocated, okay, where you have to answer 15 compulsory multiple choice questions. Okay, so meaning to say you have to answer the question and shade your answer on the OMR sheet, okay, using a 2P pencil. For section B, okay, there are 15 marks allocated, so you have to answer 2 compulsory structure questions. Okay, of course, if of each of the structure questions, there are some of the sub questions, for example, A, B, C, D, E. Okay, so you have to answer all the questions given. For section C, okay, 30 marks were allocated. Okay, so here there are 3 essay questions are given, but you just need to answer 2 out of the 3 essay questions. So bear in mind, okay, need not to go and answer all the 3 essay questions because in the paper, the instructions already given. Okay, if you answer all the 3 questions, only the first 2 answer will be marked. Okay, so this one bear in mind, do not go and attempt all the 3 essay questions. Just spend some time and choose 2 questions to be answered. Okay, and all the questions in this paper are based on the topics 14 to 19. Okay, they won't come out any question from semester 1 and also semester 2. Okay, for semester 3, okay, so our theme will be the ecology and the genetics. Okay, so there are 6 topics. Okay, topic 14, okay, taxonomy and biodiversity. Topic 15, we have ecology. Topic 16, selection and speciation. Topic 17, inheritance and genetic control. Topic 18, gene technology. And topic 19, biotechnology. Now, I'm not going to explain or go through each of the topic. I think you may refer to the syllabus. They are prepared by the MPM. And just make sure all the learning outcomes, right, they are stated or listed in the syllabus, they are mastery, okay, by all of you, right? Okay, so we'll move on to the answering techniques. Okay, for our session, I'm just going to explain to you, I give you or brief you the answering techniques for section B and also the section C only. I think section A shouldn't be any problem. Okay, okay, let's move on to the answering techniques. For section B, okay, section B is comprised of the 2 compulsory structure questions to be answered. Okay, so let's look at some of the techniques. Okay, first of all, okay, when you receive the paper, okay, so when you refer to section B, spend some time, very roughly 30 seconds, okay, to skim through the question, okay. Don't be so nervous or panic, okay, to answer the questions, okay. So make sure you skim through the question in around 30 seconds only, okay, because you have to identify the topic for the question. Because when you look at the question, look at the diagram given, okay, the diagram can be in the form of drawing, table, a graph, and so on, okay, or they can just give you a statement, okay. Then you skim through all the question A, B, C, D, E. Why? Because this will give you some idea, okay, what is the topic that is involved, right? Okay, so after you identify the topic, okay, so you recall it from your CPU or your brain, right? So you can just continue to study the diagram and labels, okay? If you are given the drawings, must study all the diagram and the labels because the question are based on the drawing or the graph or the table, right? Okay. Length and detail of the answer. These are the one normally student or the candidate, they ask, okay, how long, okay, or how many answers should be provided, okay. So the length and the detail of answer to be given are based on two aspects. Okay, first, you can look at you have to study the command terms, okay. So if the question asks you to name, so you should know how to answer name, state, explain, describe, calculate, and so on. Okay, so study the command terms. Okay, second, okay, the marks allocated. Okay, always, okay, look at how many marks were allocated for that question. Okay, so then you roughly budget how long your answer should be or how many points you should give. Of course, if some of the question they didn't limit your answer, you can write down longer. Okay, but structured question normally they just like 1 or 2 or 3 marks. So if you write too long, okay, you're going to waste your time on this structured question. So maybe you not enough time to answer the essay question later on. So you have to be careful, right? Okay, just look at how many marks allocated and you just give accordingly. Okay, you want to write out longer is okay, but just want to ask you, do you have the time to answer the other question later on, right? Okay, so this is some of the hint, okay, you can refer, okay? And next, okay, this is very important, okay? Give the exact number of answer as it is specified. Otherwise, the wrong cancel right rule will be applied. What is meant by this? Okay, now let's say there are some of the question they ask you to state one example, okay, for this one. Okay, so make sure you follow the instruction of the question. If the question asks you to state one, make sure you don't state more than one, because let's say you state 2 or 3 or 4, okay, any of your answer option is wrong, okay, the examiner will apply the wrong cancel right rule. It means if even though your first answer is correct, but your second, third, fourth answer given, okay, is wrong, okay, they will use the wrong to cancel the right. So end up you might get a 0 mark. Okay, so that's why must be careful. If the question only asks you to state 2, make sure you state 2, not more than 2. Okay, why they have this kind of rule? The reason is very simple. They just want to be fair to the other candidate, right? Use the correct terminologies. Okay, I give you some example. Now the terminologies, okay, not necessarily science terminology. Sometimes, in the marking scheme, okay, they require some of the terminologies. I mean, maybe they assume as a pre-U student, okay, you should know how to use certain terminology, even though it's not a scientific terminologies, but some of the terms, right, they require the keyword. Okay, if without the keyword or without the correct terminologies, they won't give you the mark. Okay, so and some of the terminologies, if you are using the other words to replace, they don't accept, because sometimes the word you use, they have a different meaning. Okay, I give you one example. Okay, this is one of the example. Now their candidate wrote, stabilizing selection, favors the intermediate characteristics or traits. And according to the marking scheme, the term characteristic or traits are not accepted. So what is the correct terminologies used in this case? stabilizing selection, okay, favors the intermediate phenotypes, or you want to be more accurate, in stabilizing selection, the environment favors the intermediate phenotypes. So use the word phenotype because this is required by the marking scheme, right? Okay, phenotype. Okay, don't use the characteristic or even traits, or even some candidate they use a group, or and we cannot accept, right? So these are the one you have to be careful, right? Okay, okay. Next, okay. For scientific terminologies, right? Some of these scientific terminologies, okay, you also have to be careful, especially the spelling, okay? So I give you some example. Okay, this one you learn in this semester, right? Okay, so which one is wrong? Which one is correct? Okay, this is a phylum, one of the phylum under the kingdom Animalia. Okay, so you look at the left-hand side, okay, we have this Platyhelminthes, but is it the spelling is correct? Okay, and you compare with the right-hand side, okay, and this is actually the correct spelling for the phylum Platyhelminthes. Okay, so because some of the candidates, they are not aware of this, okay, so that's why the marks are not awarded lah. Okay, even though it looks like seems like the spelling is more or less the same, but it's actually different. Okay, so the left hand side is wrong and the right hand side is the correct spelling. Okay, for the phylum. Give another example. Okay, disruptive selection. So which one is wrong? Okay, the left hand side, this is a wrong spelling, and the right hand side is the correct spelling. And these are the one very common. Okay, a common mistake did by the previous candidates. Okay, so always they cannot spell the terms, some of these scientific terms correctly. Okay, this is also very common, right? So you see the left hand side, if you pronounce the term, this is actually Klinefelter syndrome is wrong. Okay, or they spell the Klinefelter correctly, okay, the spelling, but they use a small letter for the K, and this is not acceptable, okay, because Klinefelter is the special name, okay, so that's why you have to use a capital letter, okay. Okay, sometimes the candidate, they mix up Malay terms and the English terms, so they mix it, okay, and this is also very dangerous. Supposedly it's not acceptable. You see the left-hand side, parasitisma, and this is in the Malay spelling, right? Okay, the right-hand side is the correct one, right? Parasitism, okay. Okay, for the survivorship curve, right? Okay, the type 1, type 2, and type 3 curve, okay, you should write in the Roman number instead of the Arabic number. Okay, the Arabic number is wrong. Okay, you must use the correct, just like you are writing the meiosis 1, meiosis 2. Okay, we are using the Roman number 1 and 2. Okay, so these are the small things you have to be careful. Okay, so always be careful. Don't let the examiner to have the chance to penalize your mark. Okay. About this, okay, the survivorship strategies, right, or the survival strategies, the R and the K strategies. So for the R, okay, it must be a small letter. For the K, it must be the capital letter. Okay, so the left hand side is wrong and the right hand side is the correct one. Okay, so these are the one you have to be careful. Okay, so make sure you don't let the examiner to penalize your mark because of these kind of things, right? Okay, okay. Short form, okay, for some of these so-called the common biological compound, they are accepted. Means you need not to spell the full name. Okay, need not to give the full spelling for the term. Okay, you can directly use it. Okay, like ATP, DNA, cDNA, mRNA, tRNA, H2O, CO2, F1, F2 generation, and so on. Okay, so these are the one. Okay, always shows the working. Okay, if the question requires you to calculate, okay, so make sure you show the working. Okay, so and of course the proper mathematical statement and the correct unit must be quoted, right? Okay, depends on the what kind of question what kind of unit they ask for. Okay, okay, examples in this semester, what kind of calculation you should know. Okay, I give you some example, okay, so that you are aware of it. In topic 15, ecology, remember you have to calculate the energy transfer, okay, whether it's in the form of percentage or in the form of the amount of energy, maybe in the unit of a joule or what kilojoule. Okay, based on the question, you have to depends on the question lah. Okay, and also in the same topic, for the quantitative ecology, you have to know how to calculate the species frequency, the species density, species cover, or even the relative frequency, the relative density, and the relative cover. So all this you must know the formula and you must able to calculate, right, and give the correct unit. Okay? Because many students after they calculate, yes, they find out the correct answer. Okay, they managed to get the correct value, but they forgot to put the unit or they give the wrong unit. Okay, so this is the marks will be penalized lah. Okay, and also for the topic 17, the genetics, you also have to calculate the distance between the genes using the COV formula. Okay, so all this and so on lah. Okay, these are the example of the one you learn in this semester that it requires the calculation. Okay. Okay. Now, some of the question, okay, they might ask you to sketch a graph or plot a graph, but for semester 3 normally, seldom we have the one that asks you to plot the graph lah. Normally, there's a question that asks you to sketch it. Okay. Okay. So these are some of the good grafting rules that you must obey lah. Okay. For this semester, what kind of graph they might ask you to draw or sketch? For example, the survivorship curve, right? Okay, the type 1, type 2, type 3. These are the possible the graph or the curve they might ask you to sketch, okay, in the essay question or even in the structure question. And also the 3 types of the natural selection, okay, the stabilizing selection, disruptive selection, and also the directional selection. They might ask you to sketch lah. Okay. Okay. So what are the rules you should be obeying? Right. So occupy almost the full page of the graph paper. This is if the question asks you to plot. Okay. Normally they'll give you another graph paper lah. Okay. This is the fun for the plot that asks you to plot the graph. Okay. Title of graph. Okay. Okay. If the question asks you to sketch the graph, okay. Title maybe no need, okay? No need to give the title, but the axis you must give, okay? The y-axis and also the x-axis, okay, must be correct. Okay? If you give me the wrong axis, for example, they ask you to sketch the survivorship curve, okay? Your y-axis, what should be the y-axis? Some of the students, they answer, they wrote y-axis, they label as the number of individuals, or they just label as a population size, and it's wrong. Okay, survivorship curve, the y-axis, okay, what should be labeled? It should be labeled the number of survivor, right? And it's in the log scale. Okay, so and the x-axis, some of the candidate, okay, some of the past candidate, they label it as a time, and it's wrong. Okay, what should be the x-axis? Okay, it should be the percentage of maximum lifespan. Okay, so make sure you label both of the axes accordingly. Okay, label the curve, especially when there's more than one curve in the graph, because sometimes in the graph, let's say the types of natural selection, they ask you to sketch the, let's say, the stabilizing selection. Of course, when you sketch the one, the population size, they want to show the population size against the time after, and you have to show also the before the selection, right? You must sketch the one for the population before the selection and also the population after the selection. So you sketch 2 curves in one graph. So if you didn't label your curve, so the examiner, they won't know which curve is represent before the selection, which curve is represent after the selection. So make sure you label your curve, especially when there are more than one curve in the graph. Same goes to the survivorship curve. If you go and sketch type 1, type 2, type 3 curve, okay, in one graph, of course, okay, you have to label all the 3 curves. Otherwise, the examiner, they won't know which one because they cannot read your mind. So they don't know which one is the type 1 type, so they cannot give you the marks. Okay, so these are the things you have to be careful. Okay, these all points should be accurately plotted. This is only if the question asks you to plot the graph lah. Okay, line of the best fit is also for if the question asks you to plot the graph, and you should know plot and sketch, they are different. Okay, so plot, normally you should plot on the graph paper, and normally they give you a set of data. Okay, sketch is just, you just give the correct pattern. Okay, how does the curve looks like, the shape? Okay, then should be okay. Okay, so these are the one. The last two is actually for the plotted graph. Okay, not for the sketch graph, right? And do not mix the language within a sentence. Okay, so if you want to be in English, answer in English, you answer fully in English, okay, including all the terms. Okay, don't, some of the terms you spell half in Malay, half in English, okay, and this is very dangerous. Okay, so do not mix the languages within a sentence. Write a clear and concise sentences. Okay, not need to be too long, the sentence. Okay, you can split, sometimes you can split into two sentences, doesn't matter. Okay, so make sure your sentence are clear and concise. It must represent a pre-U student's level. Okay, avoid from using the correction liquid or telah, because sometimes the candidate, they use the correction liquid. So while waiting for the liquid to dry, they switch to the other question, but then they forgot to this one. Okay, come back to the question. Okay, so these are the one. Just cross it and write the correct answer. Okay. These are the general techniques. Okay, let's move on to some of the past year question. Okay, for structure question, we'll look at 4 of them. Okay, 4 structure questions. Okay. Let's look at this. Okay, the year of 2020. Okay, question 16. Now, this question is from the topic 15, ecology. Okay, so when you first look at the question, please scan through the questions, including the diagram. Okay, so, and you have to determine which topic this question is from. Okay, because you have to retrieve the information from your brain, right? Okay, so because bio, we have to memorize a lot of things. Okay, so that's why you have to retrieve the information. Okay, so this question is on the topic 15, ecology. Is under the subtopic biogeochemical cycle. So for biogeochemical cycle, you just need to study 3 of them, focus on 3 of them: the carbon cycle, sulfur cycle, and also the phosphorus cycle. So this question, okay, read the question. A simplified sulfur cycle is shown in the diagram below. Okay, so look at the diagram. Okay, first of all, we're going to analyze, we're going to study the diagram first, okay, before we look at the question. Okay, now the diagram might be slightly different from the whatever the reference books, the notes from your teacher, and so on. Okay, but it's more or less the same. Okay, so you have to identify, okay, what is represented by all those the one you see, the K, L, and so on. Okay, now for this sulfur cycle, let's look at the K first. We are going to start from the K. Let's look at this, the compound. Okay, I think it's a compound that labeled as a K. What should be this compound? Okay. So what is actually it's actually this K is actually the main form of the sulfur that absorbed by the plants. Okay. So this is actually the main form of sulfur absorbed by the plants. Okay. The plant, they are not going to absorb the sulfur in when the sulfur is in the elemental form. I mean it's sulfur, okay, or sulfur dioxide, the one is a gas form. Okay. So plant cannot absorb in that kind of form. Okay. What kind of form the plant will absorb for sulfur? Okay, so you should know, okay, this K is actually the sulfate. Okay, this is actually the sulfate. It's same as the like the other cycle, like you studied the nitrogen cycle last time in your Form 4, and you also studied the phosphorus cycle, remember? And if you are aware, okay, plants, right, for these kind of elements, right, the sulfur, nitrogen, phosphorus, okay, plant will only absorb this kind of elements when they are in the form of like sulfate, nitrate, and phosphate. Okay, so this is not that difficult. So you have to link, maybe you can link to the other cycle and you can see the generalized pattern. Okay, plant, okay, they always absorb the sulfur in the form of sulfate. Okay, so after the plant absorb the sulfate, okay, from the soil, okay, particularly from the soil, right? What happened? Okay, of course, they're going to incorporate the sulfate to form the proteins, the plant proteins, right? Okay, and later on, after this, the animal, okay, how the animal obtain the the sulfur, okay? The animals, they obtain the organic sulfur, okay, by feeding on the plants, right, or the other animals, lah. Okay, this is represented by this arrow. Okay, after that, What is represented by this arrow? From the plants and animals, they go to this, the waste and the decaying organism. Okay, so when the plants and animal die, right? Okay, what happened? Okay, we have the decomposer. Remember, the decomposer can break down the dead plants and animals, okay, through the decomposition, okay, the decomposition process, okay, and they might release, right? What? Hey, you see, after this, there are two arrows over here. Okay, one is labeled as a step 1, the other one is pointed to the L. Okay, so step 1 is they point to the sulfate, means okay, when this decomposer, okay, they break down the dead plants and animal, okay, they release this sulfate, okay, back to the soil or even to the water. Okay, so this is happen under the aerobic condition. Okay, you must bear in mind. Okay, so waste, the decomposer, okay, they can either break down the dead plants and animals, okay, through the decomposition in the aerobic condition or in the anaerobic condition. If it's an aerobic condition, they are going to break down and release the sulfate, okay, which is the K. Okay, and the arrow point to the L. This is actually, if it's in the anaerobic condition, then they might release the hydrogen sulfide. Okay, it's a kind of gas, okay, into the soil or the water. Okay, this is if it is in the anaerobic condition. So the L will be the hydrogen sulfide. Okay, so of course, okay, look at the step 4 from K to the L. Okay, let's look at this. Okay, from K to the L. Okay, so of course, okay, this sulfate, how it can be converted into the hydrogen sulfide. Okay, actually, you have to make use of a little bit of your chemistry knowledge. Okay, from sulfate to hydrogen sulfide, this is actually the reduction, okay, the reduction reaction or the process. Okay, so meaning to say the sulfate in the soil or the water, actually it can be reduced. They can be reduced to hydrogen sulfide by the presence of this sulfate reducing bacteria. Okay, there's a presence of this sulfate reducing bacteria in the soil. They can reduce the sulfate in the soil into the hydrogen sulfide. Okay, okay, and then from the L to the sulfur and from the sulfur to the K is step 2 and step 3. These two steps are actually oxidation. Okay, because the hydrogen sulfide, okay, after we form the hydrogen sulfide, okay, it can be oxidized. Okay, it can be oxidized to the elemental sulfur, which is step 2. And the elemental sulfur can oxidize and can be further oxidized to the sulfate by the presence of the sulfur oxidizing bacteria. It can be happen in the soil or in the water, of course. Okay, so these are the explanation for this diagram. Okay, so after you understand the diagram, of course, now I explain in a long way, but during the exam, of course, you'll be very fast, right? Okay, okay, let's look at the question. A, identify the K and L. Of course, just now already mentioned. Okay, why is actually this compound K? Okay, answer, K is actually the sulfate or the sulfate ion. Okay, make sure you don't spell it wrongly. Okay, because the spelling is very important. Okay, okay, how about the L? Okay, what is this L? L is actually the hydrogen sulfide. Okay, and you don't mix up with this. Some students they spell it hydrogen sulfate and it's not accepted. Okay, so hydrogen sulfide is actually the gas, okay, that is produced by the decomposition of the waste and also the dead organism. Okay. B, state the condition which enable the process at the step 1 to occur. So as I mentioned to you just now, okay, the decomposition can be occur in the aerobic or in the anaerobic way. So for aerobic, in the present oxygen, then when the decomposer break down the dead organism, they will release the sulfur in the form of sulfate. Okay, so the condition involved here is actually the aerobic condition or in the presence of the oxygen. Okay, so actually the answer is very simple only. Yes, some of the candidates they write very long. Okay, but actually they don't have the meaning is not there. So we cannot give you the marks. Okay, so if the meaning is run away from the original, sorry, the marks will not be awarded. Question C, at which of the steps, okay, do the following processes take place? Part 1, reduction. Okay, 1 mark was given. Okay, so reduction. So just as I mentioned to you, which step? Step 1, step 2, step 3, step 4. Which step? is actually involving the reduction. Okay, so from the sulfate to the hydrogen sulfide, this is actually reduction process. Okay, so the answer, there's only one, which is step 4. Okay. Oxidation. Now, for oxidation, you see the marks allocated, okay, 2 marks. So I think, right, there are 2 answers for this. Okay, you should give 2 answers for this in order to obtain the 2 marks. Okay, so oxidation is occur when the hydrogen sulfide oxidize to the sulfur and when the sulfur oxidize to the sulfate. Okay, so if you think in the chemistry way, you look at the molecular formula, you should know this is actually oxidation. Okay, so it's very easy on it. So the answer is step 2 and step 3. Okay. Question D. Why do the bacteria involved in the step 2 are present in the shallow water and above the decaying organic metals? Okay, so be careful with this question. Okay. Okay, 2 marks were given. Okay, so why they give you 2 marks? Because one of the mark is actually for this presence in the shallow water. Why they are present in the shallow water? 1 mark. And the other mark is allocated for this. And why the bacteria is above the decaying organic metals. Okay, so that's why there are 2 marks are allocated, because 1 mark for the why they are present in the shallow waters, and the other mark why they are above the decaying organic matters. Okay, now if you are aware, hey, actually the bacteria here in the step 2, hey, they are referring to a kind of what we call it, the photosynthetic. It's a kind of photosynthetic sulfur bacteria. I think I've learned this in your SEM 1, right? The purple, for example, the purple sulfur and also the green sulfur bacteria, if you can recall, right? These are actually the photosynthetic sulfur bacteria. Okay, so they're present in the water. Okay, so and why? Can you explain why they're present in the shallow water? Why must be shallow? Okay, so why they give you the term shallow? So you should know if you are smart enough, you can guess actually, hey, why shallow water? Because this kind of bacteria, you see so-called the photosynthetic bacteria, the photosynthetic sulfur bacteria, they actually require the light energy or the sunlight for the photosynthesis. Then you're going to get 1 mark. Okay, this is the why they're present in the shallow water. And because if it's the water is in when you go deep, okay, the level of this light, okay, will be getting less and less. That's why they mentioned the shallow water. Okay, why is above the decaying organic matters? Okay, this is actually another one. Okay, so actually this is the answer because the bacteria require the hydrogen sulfide, okay, as the source of the hydrogen. Because you know these are the photosynthetic sulfur bacteria. Okay, when they undergo the photosynthesis, they need hydrogen as this one, right? They need the hydrogen source. Okay, but you should know this bacteria is different from the plants. Plants, if they undergo photosynthesis, they need also hydrogen, but the hydrogen is from the water. This kind of photosynthetic bacteria is a bit different. Their hydrogen is not from the water, okay? But the hydrogen is actually, they obtain from the hydrogen sulfide, and hydrogen sulfide is actually released by the decaying organic matters, as you study here, okay? The L, okay? So it's released by this when the bacteria decompose those the dead organism, they'll release this gas, and the bacteria, this photosynthetic bacteria, they're going to make use of this hydrogen sulfide as a source of the hydrogen, okay, for the use in the photosynthesis. Then you're going to get 2 marks. Okay, done with this question. Okay, move on to the question 2018. Okay, this is from the topic 17, okay, the inheritance and also the genetic control. Okay. Question 17, Ali is married to Fatima. They are healthy individuals. However, Fatima's father is a hemophilia patient. Now, seems like very easy, this question. Okay, so if you are aware, you should know this hemophilia is an example of a sex-linked inheritance. Okay, same goes to the color blindness. Okay, so you are assumed to know. Okay, when they give you hemophilia, or even color blindness, okay, the candidates are assumed, they're expected to know, okay, these are actually the example of sex-linked inheritance, okay. A, what is the type of inheritance disease suffered by the Fatima's father, okay? Okay. In the question, they mentioned Fatima's father is a hemophilia patient. Okay. And you know this is a sex-linked inheritance. So, then this is the answer. Okay. The type of inheritance suffer from the Fatima's father. Actually, the answer is the sex-linked or the X-linked inheritance. Okay. It's definitely not hemophilia. Some of the candidates, they answer hemophilia. Hemophilia is not a type of inheritance. Okay. So, it's an example of the disease, right? Okay. So this is a sex-linked or the X-linked inheritance. Okay. B. State the genotypes of Ali, Fatima, and Fatima's father. 3 marks are given. Okay. So, now in this question, there are no symbol given, right? Okay. So some of the candidate they ask, they said, so the question they didn't give any symbol. So what kind of symbol I should use? Okay. Now, since the symbol represent the alleles, okay, they are not given. So I think any alphabets, right, A until Z, right, they're accepted. Okay, as long as it's written as a superscript, right, of the X chromosome, because the allele is carried on the X chromosome. Okay, so you don't write on the Y chromosome. Okay, so it's in the superscript form. Okay, so any alphabet, because the question didn't give. If the question give, right, so let's say they represent by allele, let's say H. So you have to use the symbol, okay, to represent the allele, right? Okay, capital letter for dominant allele, the small letter for the recessive allele, right? So you can use any symbol, but you have to be consistent, because the question after this C, D, E, if here the B you use let's say the letter H to represent the allele, then the following question, the C, D, E, you also stick to the letter H. Don't switch to other alphabet, right? Okay, so these are the things you have to be careful. Okay, so Ali, I think I think I need not to explain. It's very straightforward. Okay, because Ali, they are healthy individuals. So Ali is a male, so that's why it must be XY, and a healthy male, there's only one possible genotype, X big H Y. So you can use others alphabet lah. Okay, so must be capital letter because dominant allele. So X big H Y. Okay. Fatima is also healthy, but Fatima's father is hemophilia. Okay, so that's why Fatima, okay, she's also healthy, but she's a carrier at the same time. Okay, because one of these allele, the small H allele, is actually inherited from her father. Okay, so her father will pass the X small h to Fatima. So Fatima must have a X small h in her genotype. So her genotype should be X big H, X small h, because she is a healthy individual, but she is a carrier, lah, right? Okay, so this is the genotype. Fatima's father, of course, because Fatima's father is a hemophilia patient. So this is her father's genotype, X small h Y. Next, question C. What are the possibilities of the son and also the daughter of the couple having the hemophilia? Okay. I think this question is this C is not related to the question on the Fatima and the Ali's the question. Okay, this is another question. Okay, what are the possibility of sons and daughter of a couple? Okay, so actually you can actually need not to construct the genetic diagram. They just ask for the possibility, okay, in the percentage or in the fraction, you can answer, okay, in the fraction form or the percentage form. Okay, let's say a couple, okay, having a hemophilia, okay, so I write in a simple way. So let's say the male, okay, is the hemophilia, okay, X small h, Y, okay. Okay, there's something, okay, okay. Right, so X small h, Y for the male, right? The male couple, okay? And the female, because she's a hemophilia as well, so X small h, X small h, okay? So you construct the genetic diagram lah. I think this one you have to construct a genetic diagram, right? Okay, so X small h, right? But need not to construct a genetic diagram in the. Okay, I think you can solve it by your own. I give you the beginning, right? So they're going to form the okay. two kind of gamete, okay, X small h and the Y. Okay, for the female, oh sorry, they only produce one kind of X big H, X small h. Okay, wait, having a hemophilia. Okay, couple having a oh, okay. Sorry, this question, what are the possibilities of the son of the couple having a hemophilia? Okay, the couple is still referred to this Ali and also Fatima. Okay, so Ali is just now, what is the Ali's genotype? Okay, Ali is a X big HY and the Fatima is a X big H, X small h. Okay, so Ali is a normal. Okay, Fatima is a normal, but she's a carrier. Okay, so you start with this, the X big HY cross with the X big H, X small h. Okay, so for Ali, you're going to produce two kind of gamete, X big H and also the Y. Fatima also produce two kind of gamete, X big H and X small h. Okay, so then you cross the gamete. Okay, fertilization occur. They're going to get four kind. I think you're going to get four kind of F1. Okay, offspring. Okay, so then from there, okay, you can find out the possibility of the son and also the daughter who are having the hemophilia. Okay, so now to answer this kind of question, you have to be careful, because if the question asks for the son, okay, in the answer you just refer to the among the son. Okay, you don't look at all the four offspring in the F1. Okay, don't consider all the four. Okay, so just look at the sons, among the son, the possibility of the son. I think there are two. Okay, so for their possible son will be produced is X big HY and also the X small HY. Okay, so these are the only two possible genotype of their son. Okay, so if they ask for the what are the possibility of the son having a hemophilia, so should be 1/2 lah, should be 50%. Okay, so the answer should be 50%. Okay, and when you continue to complete this genetic diagram, okay, for the daughter, okay, there are also two possible daughter form. Okay, the X big H, X big H, and also the X big H, X small h. Okay, so from there you consider how many percent of the daughter having the hemophilia. Okay, it should be 0%, because all their daughters should be normal. Okay, none of their daughter will be hemophilia, so the answer will be 0%. Okay, so this is the one. Okay, so you complete the genetic diagram. Okay, from the F1, you find out the possibility, but you have to be careful. Son, the possibility of son having hemophilia, you just look among the son. Daughter, you just look among the daughter. You don't mix the son and the daughter together. Okay, so these are the one have to be careful when you answer the sex-linked inheritance. The question involves the sex-linked inheritance. Okay, next. D. What are the possible genotype for the sons and the daughter of the couple? This is just based on the test, the genetic diagram, okay, from the one that you can construct just now. Okay, you just list down all the possible genotype of the son and all the possible genotype of the daughter. I think the possible genotype of the son, there are two of them. These are the answer: X big HY and also the X small HY. Daughter, X big H, X big H, and also the X big H, X small h. That's why I said the possibility of the daughter suffer from the hemophilia is 0%. Okay, so these are the one. Okay. Okay, so done with the 2018 question 17. We move on to 2017 question 17. Okay. Okay. This is from the topic 18, the gene technology. Okay. They're asking about the plasmid lah. Okay. The structure of plasmid, the PUC18, is shown in the diagram below. Okay, so this is a diagram. Okay, you can see the ampicillin resistant gene, the AMPR, and you can see the LEXZ gene. Okay, within the LEXZ gene, there's a multiple cloning site, means there's a many restriction site which can be recognized and cut by various kind of restriction enzyme. Okay, so this is from the diagram. Okay, let's look at the question. A. Name the antibiotic that can be used in the screening process above. Okay, so you should know, first of all, you must know the characteristic. What are the characteristics of a cloning vector? Okay, for example, the plasmid, Okay, so you should know one of the characteristic as a cloning vector. It should have the selectable marker gene. Okay, for example, in this case, it's an AMPR. Okay, so this is so-called the selectable marker gene. Okay, so which help in the screening. Okay, so that's why, but the question is not asked about the name of the gene. They ask you the antibiotic that can be used in the screening process above. Okay, so what is the answer? So in this case, because in the diagram, they give you an NPR gene. So the corresponding antibiotic that you can use in the screening, okay, is actually the ampicillin. Okay, so make sure the spelling must be correct. Okay, double L. Okay, so it's not AMPR. AMPR is actually the selectable marker gene. Okay, so this is the answer. B. What is the enzyme that is coded by the Lexi gene? So you should know this because you have learned this in the previous topic when you learn about the leg operon, right? Okay, so it's actually the same. What is actually the enzyme that encoded or coded by the Lexi gene? Okay, so answer A, beta galactosidase. Okay, so you don't spell wrongly the beta because some of the candidate they wrote like the capital letter B. So be careful, the beta, okay, should be there's a tail lah, right? When you write, okay, write properly the beta, okay, the beta galactosidase, right? Okay. C. What is the purpose of having the NPR gene on the plasmid? Okay, 1 mark. Okay. Some of the candidate they answer help in the screening, but I think you should elaborate more on this. Okay, so what is the purpose of having this NPR gene on the plasmid? How is going to help in the screening? Okay, so it's actually what should be the answer? Okay, to determine if the transformation process was successful, or to determine whether the plasmid had been transformed into or taken up by the bacterial host cell. Okay, so this should be the answer. Okay, NPR gene, okay, how they are, they are selectable marker gene. Okay, this NPR is actually selectable marker gene, but how it's going to help in the screening? Okay, it's going to help to determine whether the transformation process was successful or not. Because why? Because later when you do the screening, you add with the ampicillin into the culture medium, right? When you add with the ampicillin, for those host cell, the bacterial host cell, if they fail, okay, to take out the plasmid, okay, supposedly they cannot resist to the ampicillin, so they will be killed by the ampicillin. But for those host cell, if they successfully taken up this plasmid, okay, regardless the plasmid is recombinant or is a non-recombinant plasmid, okay, as long as they taken up any of the plasmid from the surrounding or the medium, okay, they supposed able to survive, okay, when you add with the ampicillin, and they'll form the colonies later on. Okay, so this is the so-called the selectable marker gene. It's actually just to determine whether transformation successful or not, or whether the plasmid had been transformed into a bacteria. Okay, and one more thing, this AMPR is not used to determine whether the recombinant plasmid had been taken, because some of the candidate they wrote to determine whether recombinant plasmid had been transferred. It's not. Okay, you want to determine whether the recombinant or this one had been taken up by or transformed into the bacteria. It's not an NPR gene. You have to look at the Lexi gene. Okay, so that is the next question lah. Okay, so NPR gene, bear in mind, is not to determine whether recombinant plasmid, okay, transform or not. Okay, it's just to determine whether plasmid. I don't care the plasmid is a recombinant or non-recombinant. Okay, so as long as there's a plasmid, then the bacteria they can survive. Okay, when you add with the ampicillin. Next, D. What happened to the Lexi gene if the gene of interest, okay, is successfully inserted within the plasmid? Means because the restriction site is within the Lexi gene, see, as you can see here. So if the gene of interest is successfully inserted within the plasmid, supposedly the Lexi gene will be disrupted. Okay, and when the Lexi gene is disrupted, what happened? Okay, so you have to mention, okay, the non-functional beta galactosidase will be produced. Okay, so these are the answer. Okay, so the Lexi gene is disrupted and non-functional beta galactosidase would be produced. Okay, Some of the candidate they mention Lexigene cannot function. Okay, it's too general because what happened if it not function? What is meant by it's not function? Okay, and some they mention the enzyme beta galactosidase cannot be produced. It's also wrong. Okay, so when it's disrupted, it still can when they undergo transcription translation, it still can produce enzyme, but the enzyme is become non-functional. It's just about the same concept as if the gene is undergo mutation, something wrong with the gene. Okay, still you can produce a protein, okay, in this case, but the protein is non-functional, okay? So this is the answer, okay? Next, E. Explain how the Lexigene is used for the blue-white screening, okay? So what is meant by this, okay? Okay, 2 marks were given, okay? So how this gene is used for the blue-white screening, okay? Actually very easy only, okay? You see 2 marks were given, right? So 1 mark is allocated for this blue, okay, the blue color, okay, the what is meant by this blue colony, okay, later on you know the bacteria, okay, some of the bacteria they are going to form the blue colonies, and some of the bacteria they're going to form the white colony. So 1 mark for each of them, okay. So how I'm going to answer this question, okay. Of course you must relate to Lexigene, okay. Read the question how the Lexigene is used, okay. First, okay, this is the first point, okay, to answer why some of the bacteria become blue, some of the bacteria colony become blue. So if the Lexigene remain intact, okay, because of the absence of gene of interest in the Lexigene, so the resulting beta-galactosidase is able to hydrolyze the X scale, okay, and this will cause the bacteria colony to become blue. Because if the meaning to say if the Lexigene remain intact, because you fail to insert any gene of interest within the Lexigene, means the Lexigene still okay, right? Means it's remain intact. So it means if when it undergo transcription and translation, it can produce an enzyme, beta-galactosidase. So with this beta-galactosidase, they can able to hydrolyze the X scale into the blue product, and therefore the bacteria eventually they're going to form a blue colony. Okay, so this is the first point. Okay, the second point is for the white. Okay, why some of the bacteria forming the white colony? Okay, so it's about the same, the sentence, but it's the other way around. If the Lexigene is disrupted because of the presence of gene of interest in the Lexigene, the resulting non-functional beta-galactosidase fail to hydrolyze the X scale, so that's why it's going to remain colorless or white. That's why this will cause the bacterial colony to become white. So this is the one you can answer for this. Hey? So after you answer the question, make sure you read again the question. Ask yourself, do you answer the question? Did you answer the question or not? Okay, how Lexigene is used, okay, for in this blue-white screening. Okay, and check with your answer again. Ask yourself, do you answer the question? Okay, if yes, then you can proceed, right? So this is the answer. Okay, for this, it's okay. You can write down longer, but not too long, because just 2 marks. Okay, it's too long. It's not worth Okay. F. What can be deduced if the transformer is white? This is very straightforward. Okay, means the recombinant plasmid had been transformed into the bacteria host cell successfully. Okay, that's why the bacteria colony will form white. The bacteria will form the white colonies. Okay, because the recombinant plasmid had been transformed, okay, into the bacteria successfully. Right. Okay, so this is the answer. Okay, move on to 2019. Okay, question 17. This is from the topic 19, the biotechnology. Read the question. Okay, the process of producing the herbicide-resistant plant is shown in the diagram below. Okay, you study the diagram and you start to answer the question. Okay, so I think you should be very familiar to this diagram. Okay, so the process is about the same as the genomic cloning. Okay, actually. Okay, but some of the terms you have to be aware, okay, because now you want to produce a herbicide-resistant plant. Okay, it will be slightly from the usual process in the gene cloning, because you have to involve the plant cell in this case. Okay, so first, okay, roughly explain to you only Okay, first what happened? Okay, you are going to isolate the target DNA. Of course, you must have a target DNA. This target DNA contains the herbicide-resistant gene. So this target DNA, which containing this the herbicide-resistant gene, is isolated, okay, from the donor cell, for example, E. coli. Okay, some of the bacteria they have this gene. Okay, so and then at the same time also, you must isolate this molecule P, which is the Ti plasmid. Okay, this is actually Ti plasmid. Okay, must be specific. Okay, from this bacteria. You have to know what is this bacteria. Okay, so make sure you know the name of this soy bacteria. It's a soy bacteria. So the Agrobacterium tumefaciens, right? Okay, so this is actually where you isolate the Ti plasmid, okay, from the Agrobacterium. Okay, then you're going to cut both of the target DNA and also the Ti plasmid using the same restriction enzyme, right, to produce the complementary sticky end, so that easy for the herbicide-resistant gene to be inserted into the Ti plasmid. Okay, then you allow the ligation to occur, where you have to add the DNA ligase, okay, in order to for the herbicide-resistant gene and the Ti plasmid to be ligated, okay, to form the recombinant Ti plasmid. Okay, so this is the Ti plasmid, okay, and then only the recombinant Ti plasmid then transform into the bacteria, okay, which is also the Agrobacterium. Of course, this Agrobacterium, its own plasmid has been removed before you use it. Okay, so we will transform into this Agrobacterium again, and you know this soil bacteria, they can infect the plant cell. Okay, so you allow, after you transform the recombinant Ti plasmid into the soil bacteria, I mean the Agrobacterium, you allow this Agrobacterium to, which carry the recombinant Ti plasmid, to infect the plant cell. Okay, so this Agrobacterium, this soil bacteria, you should know already. In nature, it is able to infect the plant cell, but in nature, it's going to cause a tumor formation to the plant. Here, of course, you already do some modification to its genome, so that you're not going to cause any tumor formation. Okay, so we are going to just make use of it to help us to, to integrate, okay, to integrate the herbicide-resistant gene into the plant cell DNA. Okay, so we're just going to make use of this soil bacteria to help us to integrate this herbicide-resistant gene into the plant cell DNA, right? Okay, while it's infecting the plant cell. Then you can grow the plant cell into to form the colors, right, in the culture medium. Okay, so then you have to do the screening on the colors using the antibiotic. And finally, after you select the colors, right, so you separate it, you have to couch it in a test tube or whatever in order to allow it to regenerate into the herbicide-resistant plant. Okay, and the technique, we call it tissue culture technique. Okay, so this is basically the process. Okay, let's look at the question. Name the molecule P, bacterium Q, and also the process T. Now, they already mentioned the P is a molecule, Q is a bacteria, and the T is a process. So they give you the hint already. So what is actually the molecule P? I think you should be more specific. Okay, so for molecule P, the answer should be Ti plasmid. Okay, this is actually Ti plasmid. Okay, because it's the one normally we use it in order to produce this the transgenic plant. Okay, not necessarily herbicide resistant, but any of the transgenic plant. Okay, you can should be Ti plasmid lah. Bacterium Q, you must know the name. Okay, so and you must know how to write the scientific name. Okay, so the correct spelling for this Agrobacterium tumefaciens, right? This is the full spelling. Or you can even give an Agrobacterium SB dot lah, right? So make sure you underline the scientific name, right? If you didn't underline the scientific name, there's no marks to be given. Okay. Process T, this is regeneration. Okay, you should know this is a regeneration. Okay. B, describe the step R and S. Okay. Now they ask you to describe. Okay, not just the name of the step. You have to give a description. But you see 2 marks were given. So 1 mark for the step R, 1 mark for the step S. You're not going to be very long lah. Of course, you are not sure you can write down longer, but just 1 mark only. Okay? Each of the steps there's just a 1 mark given. Okay, so how to describe this step R? Okay, suggestion. Okay, you can give this the step R. Okay, the bacterium Q, or actually the Agrobacterium, which carrying the recombinant Ti plasmid. You allow it to infect the plant cell, and because when it and you want to continue, it also can, because sometimes they mention infect the plant cell, and they are going to integrate the herbicide resistant gene into the plant cell DNA. It's okay lah, because you write down longer, it's okay. Okay, so this is for step R. Okay, how about step S? Okay, what happened to this? Based on the diagram, right? So what happened to this? Okay, step S, the plant cells are then grown in the culture medium and they are screened using the antibiotic, right? So these are the steps, right? The step R and step S, you must know how to describe it, right? Okay, you can write down longer, of course, but 1 mark only, right? Okay. Okay, question C. What happened to the herbicide resistant gene in the plant cell after the step R? Okay, so that's why I didn't mention this in the previous question. Okay, so this what happened to the herbicide resistant gene in the plant cell after the step R? Actually, the answer means what happened to the gene after the bacterium Q infect the plant cell. Okay, when the bacterium Q infect the plant cell, what happened? You know the herbicide resistant gene is within the TI plasmid, right? So what happened? Okay, so actually the herbicide resistant gene is integrated or incorporated into the plant cell DNA, right? So must mention the plant cell DNA lah. Okay, so don't mention plant cell. Must mention clearly it's actually the plant cell DNA. Okay, or even the plant cell chromosome. Okay, so these are the one. Okay, cannot just mention the plant cell. Okay, must mention it's incorporated into the plant cell DNA. Okay, okay. Question D. State the advantage of producing the transgenic plant. Actually, this question is very easy. You know why? Because the answer is given in the question. Okay, it's already in the question. Okay, what is the advantage of producing this transgenic plant in the diagram? So the answer, the transgenic plant becoming resistant to the herbicide. Okay, you have to base on the diagram. Okay, you don't give the other, the other advantage like what the higher productivity based on the diagram. Okay, this diagram is to aim to produce a herbicide-resistant plant. So of course, the transgenic plant will have such a the trait, right? So the resistant towards the herbicide. Okay, okay. Let's move on to the section C. Okay, section C will be the essay questions, and you should know you have to answer 2 out of the 3 essay questions given. Okay, so what are the general techniques? Okay, I give you the general techniques first before you look at the question. Okay, first, okay, so again, same as when you receive the paper and when you look at the section C, okay, when you start to answer the section C, make sure, okay, you scan through all the 3 questions first. Okay, scan through all the questions. Okay, skim through the questions in a few minutes lah. Why? Because you have to make decision which essay question you want to answer. Okay, make sure, okay, this one, because some students they just simply answer and then halfway they say, oh, I think I cannot continue, so they will switch to the other question. It's waste of the time. Okay, they cancel the question halfway and they go and answer the other question. So it's a waste of time, right? So better you skim through the question and then maybe you select the question they want to answer, do some draft, then only you write in your the answer sheet, right? Okay, so decide the question to be answered. Okay, you have to decide the question. Each of the question there's A, B, or A, B, C. So make sure you select the question, you must answer both A and B or A, B, C. Okay, don't you select the question because you just want to answer the question A. So I mean it's rugi lah, right? So you didn't answer the other question. Okay, so because each of the question there's it's a A and B or A, B, C. Okay, you must answer all the sub question. Okay, okay, answer 2 questions only, right? Write the full sentences in a paragraph form. Now, this is an essay question. Okay, always bear in mind, essay question you should write in the full sentences. For the question involving the comparison or ask for the differences, also answer in the paragraph form. Never and never answer in the table form. Okay, because if you answer in the table form, you won't give a full sentences. You won't give a complete sentence. If you didn't give a complete sentence, marks will not be awarded. Okay, so you must give a full like this. So better you answer in the paragraph form. Okay, so always full sentences. Okay, use the sentence connectors, for example, whereas or while, in order to differentiate 2 items in 1 sentence. Now, for the question which asks you to differentiate, okay, so some of the candidates, you know how they answer the question. They go and put 1 paragraph. Let's say they ask you to differentiate, let's say the amniocentesis and also the CBS. The candidate describe the amniocentesis in one paragraph and the CVS and in another paragraph. This is not the way. Okay, you want to compare both. You should write, okay, in the sentence form, and in one sentence, I want to see the difference from the two items. Okay, so this is the way. Okay, not something one paragraph on this, another paragraph on this. No. Okay, and always use the word whereas, the connector like whereas or while. Want to differentiate the two items lah in one sentence. Okay, give the diagram only when asked. Okay, some of the question like genetic. Okay, the question involve a genetic problem. If the question asks for the genetic diagram, then only you give lah. The question didn't ask for, then no need. Okay, and also like for example, the question asks you to describe the 3 types of the natural selection. But if the question didn't ask you with the aid of the label diagram, no need to give the graph. Okay, if it's just a waste of time. Okay, so give the diagram only. How they're going to ask? Normally, like for example, essay question, they ask with the aid of the graph, with the aid of the label diagram, then ask you to describe and so on. Okay, then you give the diagram or the graph, and the graph or diagram must be labeled lah. Right? Okay, all diagram must be labeled, as many as possible. You know already, scientific drawing must be labeled. Okay, so this is not an artistic drawing. Okay, whatever scientific drawing, graph, or drawings, right? Okay, you must label as many as possible. Okay. Next, for the questions involving the process or mechanism or cycle, okay, the answer must follow the sequences of the steps. Okay. So let's say they ask for the process, okay, like the process of gene cloning, okay, the process of transcription, translation, right? You must follow the sequence, of course, right? They might not give you the marks if you are not following the sequence, okay? So these are the one. Begin each of the answer on a new page of the answer sheet. So you understood why you have to do this. Okay, sometimes you finish one question, then you move to the next. Then after that, somehow in the halfway, you recall some of the point, okay, from the previous question. You want to go back, but unfortunately you didn't left any space. Okay, after that, okay, you continue with the other question. So how? Okay, so better you each question, okay, you want to answer it, okay, on a new page. Okay, so that if there any point you want to add on later on, you can just add on, right? Okay. So this is one. So make sure you write down the question number, the number of question. No need to copy the question. Just write down the number of the question that you answer, right? So, and organize your time wisely so that you have time left to check your answer, okay, for any error or omission, let's say. Okay, so these are the one. Okay. Okay, we move on to some of the past year question for this section C essay question. This 2017, this is from the topic 14. Okay, the taxonomy and also biodiversity, this topic. Okay, the question is question B, 18B, the national park in Pahang and the royal balloon in Perak. These are the example of the in situ program for biodiversity conservation in Malaysia. Explain the importance of both areas in sustaining biodiversity in Malaysia. Okay, what does the question ask for actually? 9 marks were given. Actually, the question asks about the importance, right? Okay, the importance, okay, to conserve, okay, the biodiversity. Actually, the question asking, okay, because these are the national park, the royal balloon, the state park, okay, it's actually the one of the in situ conservation, okay, example in situ conservation, okay, to conserve the biodiversity in Malaysia. So actually, this question is asked, the essay question actually asks you to explain, okay, the importance, why we need to conserve the biodiversity, okay? Because there are some candidates, I think they wrongly interpret the question. They go and answer what are the efforts to conserve the biodiversity? It's actually wrong. You see, the question asks about importance. It is not asked about the efforts, okay? So once you out of topic, then the whole essay will be 0 mark, okay? So have to be careful lah. And for this importance, okay, you see 9 marks are given. You must give the correct importance, and then only followed by the example or the elaboration. Okay, if your importance is already wrong, then no marks for the elaboration. Okay, so actually there are three importance here, the three main importance. Sometimes some students they give a lot of importance, but some of the importance they are given is actually under the same, the same point. Okay, under the same importance according to the marking scheme. So that's why you write so many, but end up you just get a very few marks on it because they're under the same point. Okay. Okay, what are the three importance? Actually, there are three importance, and each of the importance you have to elaborate because 9 marks. You give the three importance already get the 3 marks. Each of the importance you elaborate 2, maybe at least 2. Okay, 2 also you're not sure whether it's correct or not, right? So if you can, you write down longer lah, right? Okay, so that's why 3 marks plus 3 marks plus 3 marks, all together you get the 9 marks. Okay, okay, let's look at the answer. Okay, the first important, why need to conserve the biodiversity? But you focus on national park lah. Okay, this kind of the in situ program. Okay, so one of the importance, okay, to maintaining the sustainability of the ecosystem. Okay, in order to maintain the sustainability of the ecosystem. Okay, if the facts already correct, then only they have the marks for the elaboration. If your facts, I mean your importance is already wrong, okay, then they won't read your elaboration. Okay, so these are the one. So the important must be correct. Okay, so this is one of the importance to maintain the sustainability of the ecosystem. What are the elaboration you can give? Okay, okay, how to maintain, how this the national park, okay, to maintain the sustainability of the ecosystem. Okay, in terms of this, okay, for example, okay, they are involved in the protection of the water resources. Okay, you want to further elaborate, it's up to you. I don't think you need to further elaborate lah. Okay, no need to write too long, because this is already the elaboration. Okay, in terms of the protection of the water resources, you know already. Forest, okay, it's actually function as a what you call it, the water catchment area. So it's actually help to maintain the water cycle, okay, and also to regulating, to stabilizing the water runoff, okay, and also they also help to moderate some of the extreme events, for example, like the flood, the drought, and so on. And these are it's actually the elaboration from here, okay, the protection of the water resources. Also, in terms of the maintaining the sustainability of ecosystem, like national park, right, actually they also provide the protection of the habitat, okay, because national park, this national park is actually we have a minimum, we try to maintain a minimum of human intervention, okay, in order to maintain the natural habitat, okay, by providing, the many unique environments in which many of these flora and fauna, okay, they can exist, okay, and they can live in this the national park with the minimum disturbance from the human and so on. So protection of the habitat for many of the very variety of flora and fauna species, right? And also in terms of maintaining the sustainability of ecosystem, they also contribute to the climate stability through the carbon capture and also the storage, or so-called the carbon sequestration. Okay, so because national park forests, right, they act as a natural carbon sink, right? So they help in sustaining the carbon cycle, okay, and thus they prevent the climate change, the global warming, right? You want to write more, it's up to you, but I think it's enough, right? This one. Okay. Okay. The second importance you can give is to maintain the biological resources. I think this one, no problem. Most of the candidates, they get the marks because of this point only. Okay. Because they provide or maintain the biological resources, right? But you have to give some example. Okay. So what are the example you can give for under this importance, right? Think of the national park. What they can this? They can provide the food source. Okay. Of course, with some students, they ask, some of the students, they ask, teacher, this is a national park, right? So what's food source? Of course, this food source is not for the commercial purpose. If it's referred to the national park, okay? Just for your extra knowledge, this is no need to write in your exam, this one, okay? They provide the food source, but this food source for national park is not for, of course, it's not for the commercial purpose. Maybe it's for the use of those, like, in a national park is those so-called the Aboriginal people, right? The Orang Asli. They have this, normally in this national park, we also have this so-called the Aboriginal or the indigenous people village, right? So that's why they rely on the food source in this, right? Okay, so it's actually accepted. It's a food source, right? And also they also have provide these medical resources, right? Whether it's a more on traditional, or you can use it as a modern medicine, right? Okay, to sustain our life and also the health, right? Okay, and also they provide this the food or the timber products, right? So the forest, right? Okay, food or the wood or the timber products, right? Okay, so these are in terms of maintaining the biological resources. Okay, the third importance, right? We have here the maintaining the social benefits. Okay, so how? Okay, you have to give the example, right? So this is one of the importance also. Okay, so the national park, the Royal Bloom State Park, okay, they can help to maintain our social benefits. How? Give some example. Okay, in terms of what? Okay, in terms of the truth, the research and also the education, right? So because it's always this kind of what you call it, the national park, the Royal Bloom, they are actually managed or governed by some of these like the forestry department, okay, or some the universities. They collaborate with the universities and also some of the research institutions, and also with not government. They are actually they have collaboration with these NGOs. For example, the WWF and the MNS and so on. So for this, right, they have conduct a lot of this research and education, research activities and the education, and also the awareness campaign, right? Okay, from this, right? Okay, okay, recreation and also ecotourism, right? This one you know already, right? So you can actually, you can pay a visit, but you need a permit, right? This kind of a place, right? So you can do some camping, right? Jungle trekking, right? Okay, or you can even visit the Aboriginal village, right? In the national park or even in the Royal Bloom. So all these are actually under this recreation and also ecotourism. Actually, is allowed, but you have to obtain the permit, right? This is just extra. No need to write down, this one. Okay, just to enhance your knowledge, okay. Also, in terms of social benefits, there's actually this national park, or they actually provide this, they contribute to the aesthetic and also the cultural values. Okay, aesthetic, you know already, okay, you can appreciate, right, the beauty of the nature, right? Okay, when you visit this national park, the Royal Bloom, right? Cultural values is including like this what is including all those what we call it in the spiritual, the ethical values, right? That links to our human life, right? Okay, and actually the cultural value you also can refer to those Aboriginal people, right? They are staying in this. Okay, so all these are actually to maintain the social benefits under the same importance. These are the examples. Okay, so these are the suggestion of how you're going to answer this question. Okay. Next, we move on to, of course, you can write out longer. It's okay, okay? But you have to look at the time given. Bear in mind, this paper is only 1.5 hours. If you spend too long time writing so many elaborate, too long, then you might not have enough time to answer well for the other question. So you have to be, plan. You have to plan well, okay? So straight to the point. Actually, scientific essay, as I mentioned to my student, okay? Scientific essay actually need not to write too long, okay? Because we are focused on the point. As long as you have the meaning, okay, you have the point and you have the keyword, they will give you the marks. Okay, don't worry. Of course, you are not sure, you can write down longer, okay, provided you have enough time, right? Okay, we move on to the next question, 2017, question 19C. Okay, this is from the topic, I think topic 15, right? Ecology. To study the plant distribution in the forest, okay, many students, they overlook this part, okay, because they say, what this question is about, okay, so is it in the syllabus? It's actually in the syllabus, right? Okay, you see, various patterns of the plant distribution should be considered. Explain the patterns of the plant distribution. Some students, they say, oh, sir, I have no idea what to write for this, okay? Actually, this question is on the population distribution. I think if you study the syllabus, it's actually the last part of this topic. Okay, if you are aware, you go and study the syllabus again. Okay, always you double check with the syllabus. You treat the syllabus as your checklist. Okay, while we are doing the revision. Okay, as I always told my student, I say you must always treat the syllabus as a checklist. Okay, so ask yourself after you study a topic, you check, you study again the syllabus, you check one by one all the learning outcomes. Okay, so ask yourself, okay, can I describe this? Can I explain this one? Okay, so refer to the syllabus. Okay, so it's in there. Okay, the population distribution. Okay, so see, 9 marks were given. So how many patterns did you learn? Okay, the pattern of the population distribution, 3, right? So there are 3. So if you give the 3 pattern, right, you already obtained 3 marks. So, but 9 marks were given. So I assume, right, each of the what you call it, the population distribution, you have to elaborate more, lah. At least you give 2 points. So 3 marks, 3 marks, and 3 marks, altogether 9 marks, lah. Okay, okay. One of the answer, right? Okay, we have this random distribution. Okay, so if your the pattern you give wrong pattern, then no marks for your elaboration. Okay, always the same. Okay, if the facts is already wrong, there's no marks for the elaboration. Okay, so make sure you give the correct name for the pattern of the plant distribution, like this random distribution, right? So how you're going to explain it? You have to explain. Okay, the question already mentioned, ask you to explain. Okay, so it tried to link to the question because I asked about plant. Okay, so okay, in this random distribution, the plants, right, they are spaced throughout the forest, right, in a manner that is unrelated to the others. Okay, this is just elaborate what is so-called your random distribution, meaning to say all the plants that they live in the forest, they grow in the forest, they just spaced throughout the forest in a manner that is unrelated or independent to the others. Okay, so this is so-called the random distribution. Okay, why they have such a the distribution? Because of the consistent environmental resources. Means anywhere within the forest you can obtain the resources, the environment. You know already why the population they have such a pattern of a distribution. Mostly it's because of the environmental resources. Why some of the population they tend to showing a random distribution? Why some of the population they have clump? Why some is showing the uniform, right, and so on? Okay, so because of the environmental resources, okay, always is because of how the environmental resources are allocated within the environment or the habitat, right? Okay, so in this case, why it is showing the random distribution? Because the environmental resources are consistent. No need to mention what kind of environmental resources. A plant, usually the environmental resources they need to obtain is actually like light, the soil nutrients, the soil pH, the soil texture, temperature, like what else? The moisture or the humidity. These are the environment, but need not to mention, I think, right? So these are the environmental resources, lah. Okay. Okay, another one. Okay, next we have this clump distribution. Okay, so can you explain a bit on this so-called the clump distribution? Okay, why there's a clump distribution? Why is so-called this clump distribution? Because the plants are concentrated only in the specific parts of the forest. Certain plants maybe some of the plants are concentrated in the certain specific parts of the forest. Already some of the plants they need more. They need moist, and they need to live in the moist and the shady area. So they cannot expose to the sunlight. Sometimes we have this kind of plant, right? Like for example, the bryophytes, the ferns, right? They need a shady area, shady, moist area, right? That's why they'll concentrate in certain parts because of the environmental resources. Because of what? Due to the patchy environmental resources, right? So these are the one why they tend to have such a pattern of distribution. Because the environmental resources for this kind of plant, they are also patchy, right? They also focus and they're only concentrated in certain parts of the forest. So this is the one. And one more, we have this. Some of the plants, they are showing the uniform distribution, right? Because some of the plants, they are just evenly spaced throughout the forest. So this is the one. Why some of these plants they can be evenly spaced throughout the forest? Because they are due to the competition for this kind of plant. Maybe they need certain nutrients, right? They're limited within the area, within the forest. That's why they have this. Okay, they have to be evenly spaced. Okay, means they have certain distance from the others. Okay, they cannot be too close. Okay, to the other, and they maintain a certain distance. Okay, that's why this is something like in animal, you know, some of the animal, like some of the mammals, they're showing the territorial behavior, something like that, but it's not, you cannot mention the term, right? So because of the competition for the limited environmental resources, because they are competing for certain limited environmental, that's why they have to be a bit far from they have to maintain a certain distance from the others. That's why they'll tend to show a uniform distribution rather than the random distribution. Okay, because it's limited the resources. Okay, so these are, and you have more point you can give. Okay, and for this question, need not to give any example, because some of those candidates, they tend to give example of the organism they are showing a different kind of pattern. No need. Okay, no need to give example for this, because the question also didn't ask for the example. Right? Okay, next. Move on to 2014. This is from the topic 16, the selection and speciation. Okay. Read the question, question 18. Describe the bottleneck event. Okay, this is quite a popular question, right? Okay, phenomena that contribute to the changes in the allele frequencies. But this is quite popular question. Many candidates actually they answer this, but the problem is many of the candidates they couldn't score very good marks. Okay, the main problem is they're lacking of certain terms. Okay, so we cannot give them the marks. And some even they have a wrong concept. Okay, let's see okay, look at the question. Okay, bottleneck event. What is this bottleneck? When you learn this bottleneck event, you know that this bottleneck effect is actually one of the factor that will contribute or leads to the genetic drift. Okay, the so-called change in the allele frequencies is actually, you have to relate to the genetic drift. It's actually genetic drift. Okay, so you know bottleneck effect is actually one of the factor, other than the founder effect. They're going to contribute, or they're going to cause, or going to lead to the genetic drift. Okay, so actually this question asks you, can you relate this bottleneck effect to the genetic drift by referring to the surviving population? Okay, and do not confuse with founder effect, because some of the candidates they tend to confuse with the founder effect. Yeah, they go and use the term like what we call the pioneer, right? The founder migrate out from the, no, these are the one, and you want to describe the founder effect. Okay, so let's look at the answer. 9 marks. Okay, so first of all, this bottleneck effect or the event, you want to follow the question, occurs when there's a drastic reduction in the population size. You can imagine the original population size is very large, but suddenly there's a drastic reduction in the population size. Why? Don't stop here. The next point, due to the natural disaster. This is the main thing. Why there's a bottleneck effect? Can you think of the bottle? Look at the neck, it's very narrow, right? Why? Because the neck of the bottle is representing the natural disaster. They only, after the event, after the bottleneck, there's only a few, let's say, a few individuals going to survive. Let's say, so this is so-called bottleneck, because this bottleneck, the neck of the bottle is so narrow, they only permit certain individual to pass through, right? So-called survive, right? So that's why due to the natural disaster, suddenly, you see the bottle is so wide, suddenly the neck is so narrow. Why? Because there's a natural disaster. So cause a drastic reduction in the population size. And it's going to cause a random death in the population. It's random. It's by chance only. It's not something like a certain phenotype is being favored, some of the phenotypes is selected against by the environment. No, that is a natural selection. Do not confuse genetic drift and the natural selection. Don't ever mention because this phenotype is being favored, some is not being favored, is the selected against. No, that is natural selection. This is just random death. Regardless of the phenotype is being favored or unfavored by the environment, it's just a chance event. Now, only survival will contribute the genes or alleles, whatever, to the next generation. Okay, correct, right? Okay, only the survival. If you cannot survive, of course, if you cannot survive, you cannot reproduce. If you cannot reproduce, you cannot pass your allele to the next generation, right? Okay, so that's why only the survival will contribute the genes or alleles to the next generation, right? And certain alleles, right? Because you have to refer to one population. So in a population, after the bottleneck event, you're going to see the surviving population. Some of the alleles may be overrepresented. Okay, what is so-called overrepresented means the allele frequency will increase. Okay, if you compare to original, okay, you compare the surviving population and the original large population, you're going to see some of the allele maybe the frequency going to increase, right? Overrepresented. Underrepresented means maybe some of the allele before this is very low and very high, the frequency before the bottleneck. But after the bottleneck event, decrease drastically. Okay? So that's why it's decreasing the allele frequency. Or some even totally eliminated. Okay? All these are due to chance only. Okay? So these are the point you can give, regardless of whether the allele is beneficial or harmful. As I mentioned to you already, it's not something to do with whether the phenotype is being favored or unfavored, whether they can adapt it to the environment. No, nothing to do with that. That is natural selection. Okay, so this is all by chance. So it's regardless whether the allele is beneficial or harmful or the kind of defective allele, right? Okay, so because who is going to survive is just a chance event. Okay, not because of the survival. Because why the person can survive? Because he carries an allele which is favored by the environment. No. Okay, so this is the one. Regardless whether the allele is beneficial or harmful, and this will result in a change in the gene pool. Okay, when the allele frequency change, of course, the gene pool is also not going to be static. Okay, it's going to be changed. Okay, the composition of the gene or the alleles, right? Okay, because the frequency change, right? So that's why there's change in the gene pool of the surviving population, right? What else you can mention? 9 marks. Okay, other than that, you can mention, of course, okay, try to relate to genetic drift. Relate how the bottleneck leads to the genetic drift. Of course, first you have to know what's meant by genetic drift. Okay, so of course, the smaller the population, the greater the effect of the genetic drift. Okay, so these are the one you have to know. And also, when this bottleneck event occur, of course, they're going to reduce the genetic variation within the population, and leads to the genetic drift, right? Okay, reduce the genetic variation also. Okay, and finally, I have this point. Okay, you also it might lead to the formation of the new species or speciation process over a long period of time. Okay. Always genetic drift, you know already, genetic drift is actually one of the factors that involved in the formation of new species, but it takes time. Okay? It's going to take very long period of time. Okay? Then only they might form a new species. Okay? So these are the points you can give. Okay? So after you answer the question, always check with the question again. Ask yourself, okay, have you answered the question? Okay? Have you described the bottleneck event? Have you relate how this bottleneck event contribute to the changes in the allele frequency? If yes, then it's safe, right? Okay, double check again with the question. Maybe you miss out some points, right? Okay, so these are the one. Okay, we move on to 2021. This is from the topic 17, the inheritance and also the genetic control. Okay, it's a huge topic. Okay, I want to discuss this question, 2021, question 28. In the P plan, okay, so read the question, because many students, when they read the question, it's very long, they tend not to answer it. Actually, the question is not that difficult, okay? But as I said, sometimes the student, they tend to, memorize the notes given by their teacher or the one from the books or reference books. They cannot, let's say the question asks them to solve, let's say, because this topic, genetic, they can either ask theory question or they can ask you to solve a genetic problem. So you have to be flexible sometimes, right? Okay? So you have to know how to apply your knowledge, right? Okay, so in the solving the genetic problem. So look at the question, read the question first. Okay, in a pea plant, the purple is the dominant trait. They didn't give you any symbol to represent the allele, you see? That's why later on, if you want to use a symbol, you have to denote first. If let's say you want to use a symbol, okay? So, but this question, they didn't give any symbol, right? Okay, the purple is the dominant trait and the white is the recessive trait for the color of the flower. Okay, then ask you to explain a suitable procedure of crossing the plants to determine the genotypes of the purple flower. 7 marks were given. Okay. Now, many students actually they have no idea what the question is about actually. Okay. When they see explain a suitable procedure, they already panic because they thought this is a what kind of experiment is this? But this is just the one you already learned, the kind of genetic cross. Okay, it's the procedure is also not the one you have step 1 is what, step 2 is what. So it's actually just ask you to describe a kind of cross you're going to use to determine the unknown genotype of the individual they are showing a dominant phenotype. You know, this purple flower is dominant trait, right? And let's say I give a symbol, let's say, okay, purple is a P. Okay, let's say the P is the allele to represent the big P. Okay, for the purple color. Okay, so you know, if I give you a purple flower, you know the genotype of this purple flower can be either a big P, I mean the homozygous dominant, or it's a heterozygous, a big P, small p. Okay, that's why how to determine this whether it's a big P or big P, small p. I think you learned it before, right? Okay, how to determine if I give you a dominant phenotype? Okay, how you know whether it's a homozygous dominant or it's a heterozygous? Okay, of course, you need not to apply this if I give you a white flower. Why? Because white flower, automatically, you already know it must be, the genotype must be small p, or homozygous recessive. That's why this cross, we're not going to use to test the genotype of the individual that's showing the recessive trait, because it's straightforward one, no need to test it. Okay, so what is actually the cross that involved here? So it's actually the test cross. Okay, then you have to explain a bit how you're going to do the test cross. In this case, okay, apply to this. You have to make use of the information from the question. Okay, so don't shock sendiri, right? Okay. First, answer the question first. Okay, now I give you a purple flower. Okay, so you know that the genotype of a purple flower. Okay, it can be homozygous dominant or heterozygous. Okay, so you mention first. Okay, some candidate they ask, some student they ask, can I use the symbol? Okay, you want to use the symbol like the big P, small p, you have to denote the symbol first to be safe. Supposedly you have to denote the symbol. Let's say you mentioned let the big P represent the allele for the purple flower, and while the small p is represent the allele for the white flower. So something like that. Okay, then you can use it after that. The genotype of the purple flower can be homozygous dominant in a bracket big P, or heterozygous in a bracket big P, small p. It's up to you lah. Okay, but if you want to use the symbol, you have to denote the symbol first lah. Right? My advice lah. This is my advice. Right? Okay. Then point number 2, a test cross. Right? It can be carried out. Okay. Of course, you have to mention what is the cross. There's a mark for it. Okay, a test cross can be carried out in order to determine the unknown genotype of the purple flower. Okay, can you please elaborate further on this? Because 7 marks were given. Okay, so the P plant with the purple flower, now the genotype is unknown, right? Okay, so how are you going to do this test cross on this purple flower? So we are going to do this. Okay, how? You have to further elaborate. So the P plant with the purple flower with the unknown genotype is crossed with the P plant with the white flower, which is a homozygous recessive. Always, you want to perform the test cross, you must cross it with the homozygous recessive. All right. Okay. How? You can further elaborate by transferring the pollen grains of one plant to the unpollinated stigma of another plant. This is the way actually. Okay. You transfer the pollen grain of one plant to the unpollinated stigma of another plant. Okay, either one, right? You can you transfer the pollen grain from the purple flower to the white, or vice versa. It's up to you. So that's why I didn't mention. Okay, it's either one, right? Okay. Then wait for the fertilization again, so on. They produce offspring. Okay. So point number 5. Okay, you can mention, right? The unknown genotype can be revealed. Okay, based on what? Okay. Based on the phenotypic ratio shown by the offspring from this test cross. After you obtain the offspring, you might obtain a lot of offspring. Okay, so you study the phenotype of all your offspring. Then you come up with the phenotypic ratio. Right? Okay, offspring from the test cross. Okay, based on the phenotypic ratio shown by the offspring of the test cross. Okay, then point number 6. So what are the possible phenotypic ratio? There are 2 possibilities on it, right? Okay, just write down. If, use the word if lah, because we are this one, right? We'll make this one prediction. Okay, so if all or 100% of your offspring from the test cross, they are purple, okay, the one you observe is purple, they are purple, all of them, then you should know the unknown genotype is a homozygous dominant. You can construct a genetic diagram to test on this side, but if you are the one already you studied well, you should know if 100% is purple, then you should know the unknown genotype is also homozygous dominant. Okay, automatically you should know. Okay, but you can draw the genetic diagram if you want, right? Okay. And another possibility you have to mention also, but if the offspring are showing 50% purple and 50% white, then you know the unknown genotype is heterozygous. That's it. Okay. So after you finish your essay, check again with the question. Did you answer the question? Have you answered the question? Okay. So this is the one. Okay. You can write down longer. I give you 7 point only one. Right. So you can write down longer if you have an extra point. Right. Okay. So no problem. Look at the time. Okay. I'm afraid you not enough time later on for the other question. Okay. Next, we move on to 2015. Right. Okay. Okay, this is from the topic 18, the gene technology. Question 19a, explain the production of protein from the mRNA using the genetic engineering technique. Okay, you read the question. Some of the candidate, I mean the weak candidate, when they read this question, they didn't read the question, they didn't study the question well. They explain about translation. And you know it's wrong. Okay. I know when you read the phrase like production of protein from the mRNA, you think of translation, but it's not. This question is totally not related to translation because you see, using a genetic engineering technique. So this question is actually asking about the cDNA cloning. Okay, because cDNA cloning, you know, we have 2 kinds of gene cloning, right? The cDNA cloning and also the genomic cloning. So this question is actually cDNA. Because why? Because they already give you the hint, right? You are using the mRNA as a starting material, right? Okay, so mRNA as a starting material, that is a cDNA cloning. Okay, if you are using the DNA as a starting material, that is a genomic cloning. Okay, so actually you have to describe the cDNA cloning. 11 marks were given. Okay, so you have to explain the step lah. Okay, the step. Okay, so you should start with the mRNA isolation. Okay, so let's look at the first point. Okay, you can write like this, right? mRNA is isolated from the donor cell. Okay, because the question didn't give what kind of the gene of interest that we want, what is the donor cell. Just mention donor cell in general. Okay, so always read the question. Okay, the information you should get it from the question. Okay, so since there's no information on this, I just write donor cell. At the same time, the plasmid is isolated from the bacteria. You know plasmid, okay, cloning vector. Okay, you must isolate it from the bacteria, right? Okay, after you isolate this mRNA, after you isolate the plasmid, what are you going to do on the mRNA? Okay, you cannot directly insert this mRNA into what the DNA. No. Okay, so what happened? What you're going to do? You have to add with the enzyme. What kind of enzyme you have to add it to the medium? Okay, so first you have to add with this reverse transcriptase. Why? This enzyme going to synthesize the single-stranded cDNA by using the mRNA as a template, right? Okay, so synthesize the single-stranded cDNA by using the mRNA as a template, and this process is called reverse transcription. Okay, so after that, you have another enzyme, the DNA polymerase or the DNA polymerase 3, right? They're going to synthesize the second cDNA strand. Thus, they're going to form a complete, the double-stranded cDNA. Now, after you form a double-stranded cDNA, then only both the cDNA and the plasmid, and then you can add with the restriction enzyme already into the medium so that both the cDNA and the plasmid are cut using the same restriction enzyme. Why? To produce a complementary sticky ends. Okay. What next? Okay, after you cut it, okay, insert lah, right? So that the next step, you allow it to insert, you allow the gene of interest is inserted into the plasmid, right? But you know it's not easy, right? It's not easy, okay, to be inserted. When most of the time we fail to be inserted into the plasmid, but you need an enzyme here. Okay, when you insert the gene of interest into the plasmid, they need an enzyme. Otherwise, it cannot be, they cannot anneal together. They cannot stick together, right? Okay, so both the gene of interest and also the plasmid, they are ligated by using the DNA ligase. This is a next enzyme. We have to add you to the medium also to form a recombinant plasmid. Okay. After you form the recombinant plasmid, then only what you're going to do? Okay, the recombinant plasmid will be transformed into the bacteria. Okay, so before I continue, please use the correct terms, the proper terminologies to describe the step. Some students, they mention put in, the one join together, right? So try to use the proper term like ligated instead of join, right? Okay, so these are the terms that actually the examiner want to see, right? Okay, it's transformed into the bacteria. Okay, of course, the rate of success rate is very low on it. That's why you have to, after your transformation happen, after it, you have to do the screening. Okay, the bacteria are screened by culturing them in a medium containing the two substances, right? The antibiotic, for example, ampicillin, right? And also the XKL. So the method, we call it the blue-white screening, right? And positive clones, I mean the white colonies, right, will be isolated and they'll be cultured separately in a fermenter in order to amplify the recombinant plasmid and also the synthesize the protein of interest. Because the question didn't mention what kind of protein, so I just mentioned the protein or the protein of interest. Okay, so and finally the protein can be extracted and purified. This is just an extra point. Okay, so so this is how the production, how you produce a protein. You read the question again. Have you answered the question? Explain the production of protein from the mRNA using the genetic engineering technique. Okay, see, this is not a translation process, but then some of the candidates, you see, whole page they write, they describe the translation, then 0 mark. I cannot give them a mark, any 1 mark also, there's no, because it's wrong. Okay, so that's why this is actually to describe the cDNA cloning. Okay, last question, we have this 2020 from the topic 19. Okay, question 20B. Describe how the PCR-based DNA fingerprinting is used to identify a suspect in a criminal case. 5 marks only. Okay, 5 marks only. But you, of course, you can write out longer. Okay, so because we do know the marking scheme, so you have more points you can add on, right? Okay, doesn't matter. Okay, so but you see they mentioned PCR-based, so I think you must mention a bit on this PCR. Okay, what is the use of PCR in this DNA fingerprinting? Okay, now they use the DNA fingerprinting here. They use it to solve a criminal case. So how you're going to do so? Okay, point number 1. Of course, first you must obtain the DNA. The DNA must be extracted or isolated. You don't mention take out from. Okay, use a proper term. Then some candidate they use take from. Okay, obtain from. Can you use a proper word like extracted, isolated? From the tissue sample they are obtained from the crime scene, the suspect, and also the victim. Okay, you must obtain the tissue sample must be obtained from not only the crime scene, suspect, because later on you have to compare. You compare the DNA they extracted from the tissue sample obtained to the crime scene. Okay, compare with the suspect, compare with the victim, because we do know maybe the DNA is belong to the victim, then the result is not valid because we want to find out who is the murderer, right? Okay, so you must this one, okay, but all you must compare. Okay, compare, doesn't matter, you can this one. Okay, tissue sample, for example, you can have a blood, hair, whatever, semen, right, skin, and so on, right? Okay, but extracted from the tissue sample. Don't ever mention the DNA is just collected directly from the crime scene, because there's a candidate they mentioned DNA obtained from the crime scene. No, it's not. It's not the DNA is on the floor. It's actually extracted from the tissue sample. They are obtained from the crime scene. So make sure your concept must be correct, right? Point number 2, you can use a polymerase chain reaction, the PCR method. Okay, this is not to analyze the thing. PCR is a method to amplify, not to analyze. Okay, just to amplify the specific region, the specific target region of your DNA. Okay. Okay, after you do the PCR, right? So you already amplify many copies of the DNA. Okay, then only you can use the DNA, because sometimes the DNA extracted is very small amount. Don't worry, you can always use the PCR to amplify it. Okay, Then only after you amplify the DNA will be cut into the fragments from all the samples. Cut into the fragments by using the same restriction enzyme. Then after you cut the DNA into fragments, right, using the same restriction enzyme, then the DNA fragments, you let them to be separated on gel electrophoresis, right? This one based on the one, no need to mention, it doesn't matter. Based on the size of DNA, charge of DNA, pore size of gel. So most important to mention is separated by the gel electrophoresis. Must separate it. And then, you're going to do this, the hybridization, sorry, not hybridization. This is a Southern blotting. Do the Southern blotting first. The DNA fragments, then they are denatured into the single strand, right? Because you immerse it in the alkaline solution, right? You let them to be denatured into the single strand and to be transferred to the, maybe it can be the nitrocellulose membrane, whatever, nylon membrane or whatever this one, but process is called the Southern blotting, so this is the, after you denature into single strand, the single strand, they will be transferred to the nitrocellulose membrane. Then you remove the paper blot. Then you put it in the plastic bag. They contain the solution with the radioactive DNA probe, because the radioactive DNA probe, they'll bind to the complementary base sequences on the DNA fragments. Because the radioactive DNA probe is also single strand. So they try to bind with the complementary base sequence on the DNA fragment, which is on the paper blot. But of course, not going to be all the DNA probe will be bind to the, the complementary base. Some is not going to bind, right? Don't worry, because after this step, you can rinse away those unattached punya probes. So those unattached DNA probe will be rinsed away after this. Then what next? You can do the autoradiography. Just lay a film of this X-ray film on it, and then you perform the autoradiography, and you're going to make the DNA fragments visible. You're going to show all the DNA fragment bending patterns. And finally, after you obtain the DNA profile, I mean the one with the DNA fragment bending patterns, then you can compare between the what you call it, the tissue that the DNA that you extract from the tissue sample from the crime scene, all those suspects, and also the victim. See, it belongs to who. Which one is a closely matched, then perhaps that is the murderer, right? So this is the concept. So these are the after you answer again, you check with the question again. Do you answer the question? Explain the what you call the question. Describe how the PCR-based DNA fingerprinting is used to identify the suspect in a criminal case. So already answered, then should be okay lah. So this is the one. Okay, I think that's all from me. Wish you all the best on your upcoming exams, and also I hope all your hard works will pay off.
Thank you. Terima kasih diucapkan kepada Mr. T di atas perkongsian yang cukup jelas pada hari ini. Tidak lupa juga ribuan terima kasih kepada pelajar-pelajar dan guru-guru STPM 2023 yang mengikuti program kita pada malam ini. Sebelum itu, saya minta pelajar-pelajar untuk mengisi kehadiran pada hari ini. Klik pada link yang disediakan atau scan pada QR code yang dipaparkan untuk kehadiran anda. Untuk makluman, program ini akan diteruskan lagi dengan mata pelajaran Matematik T pada 22 Mac 2024 pada hari Jumaat jam 2.30 hingga 4.30 petang di platform Zoom dan YouTube. Jangan lupa klik link pendaftaran untuk mendaftar. Seterusnya, jangan lupa juga untuk follow media sosial kami di Instagram, Facebook, TikTok dan subscribe channel YouTube YPC International College untuk mengikuti seminar yang seterusnya. Baiklah, itu saja. Terima kasih dan selamat petang semua. Jumpa lagi pada program yang akan datang.